Good afternoon. We're going to talk a little bit about next week's midterm. Normally I'd be talking a little bit more about the labs again and some lab report tips, but I think at the moment it's more important today that I talk about next week's midterm in terms of the formatting and a few things around studying and all that, and then we're going to start topic 6. All right, so let's talk about that midterm. Oh, that's not the midterm. That is for another class. Sorry about that. Anyway, let's talk about that midterm. Here's a little joke from the book, Through the Head and Gone Forever. Hopefully that is not going to be your case for the midterm. So midterm 1 is next Friday. It's 1 week from today at 4:00 p.m. And the midterm is going to be online. I was hoping we were going to be doing in-person midterms, but they've said nothing like that is going to happen until possibly after the reading week. So I apologize. I understand the situation is not ideal. We are going to be using something in the midterm called Lockdown Browser, and I will be talking about that in a moment. So Lockdown Browser, this is a system that basically doesn't allow you to use any of your other resources on your computer. So the whole idea is it's kind of an artificial proctor, an artificial exam monitor. So here are some tips for using this. First of all, it's important that you treat this as an in-person exam, meaning you should be studying and you should not be having access to any notes or anything like that. So treat it like any other exam except for you're doing it on the computer at home, presumably. The lockdown browser will monitor you, meaning it will expect you to have a webcam on your computer. And it's going to basically watch you and record any suspicious activity. So in order to access it, you need to log into Moodle through Chrome or your usual browser to access the test. And if you don't have it already on your computer, it will install at that moment. I put a sample quiz up on Moodle, and I'm hoping everyone will have had a chance to try it out to make sure that your technology is working properly. And basically what I will do is I will email you next week if you haven't tried it out, just to remind you to try it out. I am not responsible that if your technology failures, if your internet is bad or something like that, then you need to get all that sorted out before Friday. Something doesn't work during the test, reboot your computer. In fact, I recommend that you reboot your computer, about 15 minutes before you do the exam, just so that it's all ready to go and all fresh. But if you're having problems during the exam, email me as soon as possible. I will have my exam going. One thing I might do is actually make sure I'm on campus so you can actually phone me, but I'll have to see if I can do that next Friday. So to show your ID, if this was a final exam, I would ask you for your ID, but I know what everyone looks like, so don't worry about showing your ID. But it is going to ask you to show your ID, and it's going to take a photo of you and all that. But do make sure you do the sample test, and do so as soon as possible. And I will send a reminder on Monday. That's just tomorrow, but by tomorrow I mean next business day. And give that a try, and you'll see what it's all about. I will also my plan is on Monday to send out some instructions regarding the test so that you have a bit better of an idea. I just didn't have time to get that together today. But if you've used Lockdown Browser before, then you'll probably have an idea what this is all about. So let's talk about the midterm in particular and what it's going to look like. It's going to be 35 marks. And I'm not sure if this is going to be the final format yet with the online. Sometimes I tweak things a little bit, but this is generally the format of my exams. I have some multiple choice and true false worth about 15 marks. Usually they're kind of jumbled together, not necessarily separate sections. Multiple choice and true false are kind of the same type of question. True and false is maybe easier because you have A or B, whereas multiple choice you have 4 or 5 answers. There are going to be some short answer questions. There are going to be there's probably some matching or one matching question or two, depending on what they're worth, and then some longer answer written type of questions. So this PowerPoint, by the way, I'm going to go over it in detail next Wednesday during a review session. If you want to look ahead, you can see some of the sample questions. Today, I'm just going to be talking a little bit about studying and come back to this on Wednesday. And Wednesday will be a full review session. So by the way, we covered a lot of material We have covered 7 topics. Well, we've covered 6 so far. We're going to cover the last topic today and on Monday. So it's really important that you start studying now. We've covered tons and tons of material. And it's important that you know this material well. I know some of the stuff you've seen before, probably in high school and whatnot, but you do need to know it thoroughly. And so I want to talk a little bit about studying and what you can do to prepare for this. So studying, this is obviously just a joke. It says the act of texting, eating, and watching TV with an open textbook nearby. That's not going to work for you. You need to get stuff into your brain, and you need to perform memories, and then you need to be able to take it out of your brain and put it onto a test is what you need to do, whether it's on paper or a computer. This is what you're doing. So here's some tips, right? Review the lecture notes. I am going to use the lecture notes to form the basis of my tests, okay? And so if there's something I didn't cover that's in the textbook, it's probably not on the test. You can always ask me too, if you're not sure. So start with your notes and then start filling in missing pieces. You can use the textbook, you can use the PowerPoints, internet. There's lots of places you can fill in any missing pieces. Always a good place to start. Review the study questions I have at the end of each unit and the vocabulary words. That is often where I start when I make my tests. I look at those study questions and think, I could make an exam question around that. And that's where I'm going to go. It's really important to make notes. If you want to form long-term memories, notes is a very important strategy. And I'm going to replay that video that I showed you in the very first class that talks about making notes. So I'll get to that in a second. You can make use of my office hours. I know virtually it's kind of hard, but you can just schedule something with me. I'll meet you anytime I'm available, whether it's during my office hours or not. If you are somebody who likes to get a little help, there is help on campus. We have tutors at the Academic Success Center. Dana is very good with biology. You can talk to her. You can also talk to a learning strategist. They are in the Accessibility Services Department, and they deal with people with disabilities, but they're not just for people with disabilities. They're for anybody, and they can give you study strategies and talk about what you've done and kind of help you to learn how to make use of making mind maps, or if you have a hard time understanding how to do multiple choice. These are the people. You can make an appointment and make a meeting with them. They're excellent meetings. You also notice I have a sample midterm and practice multiple choice questions on Moodle. Do those, go over them. And if you can do well on those, it's going to help you a lot to do well on the midterm. So like I said, just keep in mind of how you're studying. You're doing constructive things that are forcing you to organize and do those kind of things. That's going to help you to get in your brain. If you're just being passive, just watching the lectures or just reading the notes, you're probably only going to get like, 10, 20, 30%, right, of that information in. But as you get to be more constructive, it's going to help you form memories. So I'm going to replay this video for you. I played it on the first day. It's just 2 minutes long, and it talks a little bit about kind of a systematic way to studying biology. And I really think that this is one of the best methods. So I will play this for you right now, and hopefully it's a recap on this. And if you want to play it again, you can check out the link. It's found on these slides. I have them posted on Moodle. Just scroll down until you get to the midterm 1 review part. Okay, so that's kind of my tips for now. I'm going to come back and talk, like I said, Wednesday is going to be the whole session on Wednesday is going to be review. So I'm hoping between now and then you've had a chance to do some review and you can bring out some good questions and whatnot. And like I said, take notes. I'll show you, here's an example of some notes done by a previous student in this class, and this guy did very well. His name is Malaz, and he was gracious enough to share his notes with me. And he was showing me what he did. And I thought it was kind of an interesting little system, is he would take each topic in Biology 107. And basically his goal was to kind of, he made his initial notes, and then after he made his initial notes, what he did was basically make a one-page summary of all of that topic. So in Biology 107 for midterm 1, he had 6 pages. So by the time he kind of got to that stage, he actually knew a lot of this material already, he found. And he had a lot less to study, 6 pages, as opposed to, I don't know how many pages of notes we have now between topic 1 and topic 6. You're probably looking at 60 or more pages easily. And anyway, just to give you some ideas here, there are many, many suggestions online, but I'm a big believer in taking notes because it forms memories and forces you to organize things in your mind. Like I said, don't forget to practice, do the practice exam and the practice test questions. They're the multiple choice questions. They're going to help you a lot. Okay. If there's any questions, please do throw them in the chat. Like I said, I'm going to talk more about the midterm on next Wednesday. We're going to talk about everything on Wednesday, the whole review session. We still have topic 6 to get through. So we talked about membranes, we talked about viruses, we talked about bacteria. The last kind of piece to talk about before the midterm is eukaryotic cells. And so all those organelles and those kind of things. So let's get into that. Have some notes, paper ready, because we are going to take some notes about the different organelles and whatnot as we get there. And so we'll get into this and talk about eukaryotes. So remember the biggest difference, we made some notes last unit about the differences between eukaryotes and prokaryotes, and we'll probably come back to that table again at least once somewhere near the end of the semester when we've talked about other things. But of course, the big thing is that a eukaryote, U stands for nucleus, or U stands for true, and karyote stands for nucleus. So that's the big thing, is they have a nucleus. So here's actually a eukaryotic cell. This is a white blood cell, and you can see it's attacking those little bacterial invaders. I don't know what these projections are called. They could be pseudopods, but I think since they're really skinny, they actually have another name. I'd have to look that one up. So this is kind of, I guess, yeah, this is pretty much similar to the table that we made in lecture. You can see the examples there. The prokaryotes include the bacteria and the archaea, and the eukaryotes include the animals, plants, and the protists. I guess there's a couple other pieces of information. Maybe we didn't mention typical size, dimensions, those kind of things. Do they have a nucleus? Yes or no? What kind of cell wall they have? A little bit about their DNA, of which we're going to come back and talk about DNA a bit more. Like I said, we've got these histone proteins, which is something we haven't talked about yet. Histone proteins are right here. We're going to talk about those in topic, I think it's topic 10 or something like that. And we talked about ribosomes, and we're going to talk about mitosis and meiosis in topic 10 and so on. And we haven't talked too much about flagella. A little bit about the bacterial flagella, and we're going to talk more about eukaryotes actually next week in the lab, and as well as in topic 7, so kind of a little bit after the midterm as well. So here's kind of your textbook picture of all the different types of cells, and very, very basic in some way. You can see there's an animal cell and a plant cell, and so that's something we're going to start to distinguish between today. And there's our prokaryote, and so now you know all about a prokaryote. In that, the last couple of lectures we talked about things like peptidoglycan, capsules, slime layers, pili, those kind of things. And so today we're going to get into these eukaryotes. So the first thing to talk about is the cytoplasm. And if you remember with E. coli, I was telling you the cytoplasm is not just a bunch of liquid, right? The cytoplasm is full of enzymes and solutes and things like that. And really, this is the site of chemical reactions. And of course, the organelles are going to be found in the cytoplasm as well. So we'll make a note on that in a second. I just want to take a look at the next slide here and show you. And this is kind of just a hint on some of the other things we're going to see in the cytoplasm that we're going to talk about in topic 7. Something called the cytoskeleton. So these are protein fibers. Okay, so here we go. We're going to make some notes like this. Like I said, usually I do this on the whiteboard. On the computer, it's just easier to type it in than try to write it in. And we're going to talk about all of these different structures. So if you take a look, there's the first structure, and we're going to talk about the function. So the cytoplasm, right? So the cytoplasm is a gel-like fluid. There we go. And what does it contain? It contains solutes, enzymes, contains the organelles, and the cytoskeleton. Okay, so don't worry about filling in all those other things. We're going to fill them in as we go along. As you can see, the next thing we want to talk about is a whole bunch of things around the nucleus, okay? And so we'll come back to this here in a minute. But let's talk about the nucleus. We've got lots to say about the nucleus. The nucleus, of course, is that big blue blob in the middle. And but before we talk about the nucleus, one thing I just want to say is we have all of these organelles. And these organelles are, most of them are kind of interconnected, right? So if you take a look at the nucleus, like I said, we'll come back and we'll talk about the nucleus here in a minute. There's the nucleus way over there. And notice we have ER, that's Endoplasmic Reticulum. And those two things are actually connected directly. Then we have these little blobs here, those are called vesicles. And they connect the ER and the Golgi body. And the Golgi body is connected to the plasma membrane in the same way. We have these lysosomes. And so we have all of these things in there. And they're often called the endomembrane system. So endo means within, okay? So these are the membranes within a cell. So one thing to note, and we're going to kind of come back to these next days, this does not include the mitochondria and chloroplast. Don't worry about them. We're not going to get to them until Monday. So that's next week's problem. There's another picture, by the way, of the endomembrane system. I like this one a little better because its size is very accurate in terms of the organelles, and it's actually a plant cell. So you can see some features in there you wouldn't necessarily see in an animal cell. Like you can see the central vacuole is right here, and you've got oil bodies and things like that you wouldn't see in an animal cell. Okay, let's talk about that nucleus. So most people know that that is where the DNA is found, the chromosomes. So that's good, okay? That's kind of the basis, the place to start. This is the place that holds on to your genetic material. The nucleus can be very large in some cells, up to 10 to 20% of the cell volume. Sometimes it's a little smaller. Plant cells tend to be quite large, so the nucleus is more like 5% of the cell volume. But it is quite large, and there it is represented there in the blue. Here's the image from the textbook. And you can see the chromatin is in there. I think I have a definition here for chromatin. So chromatin, chromosomes, these words are kind of used interchangeably, and I'm not going to distinguish the difference now. But notice that what they are, right, is DNA plus proteins. So chromosomes is DNA plus proteins. And those proteins have a name. They're called histone proteins. We're going to talk about them a little bit more in topic 10. But basically, they're there for packaging the DNA. The amount of DNA in a human cell is like something like 1.5 meters of DNA or something like that. I can't remember the exact value. But if you think about it, the cell is like about 50 micrometers. So that's a lot of DNA to package in a teeny tiny little thing. So you got to package it in there so that it does not get all tangled up. And that's the whole idea behind these histone proteins. So there's the DNA. That's kind of important, all about the genetic content of the cell, and we're gonna talk a lot about that in topics starting at topic 10, and we're gonna talk about DNA like a lot in the last part of the course. So we'll get there eventually. One other thing that is in the nucleus is something called the nucleolus. Okay, so the nucleolus you can see is this sort of dark patch, And so what is going on in the nucleolus? I don't know if I have it on the slide here. Yes, I do. The nucleolus is basically where ribosomes are put together. So the reason why it's dark is because it stains really well, and what that is ribosomal RNA. And that ribosomal RNA stains really well. And so again, ribosomes, we're going to talk about them a lot when we get to topic 14, and we're going to talk about a lot about translation. So maybe that's topic 15, doesn't really matter. Later, we're going to come and talk about ribosomes. Come back and talk about the nucleolus, okay? So kind of looking at that nucleus now, the nucleus is surrounded by a membrane. It's called the nuclear membrane. And in fact, it's a double membrane. Take a look at this picture right here. Look at that. See, the membrane kind of goes here, and then it folds in on itself. And so it's actually 2 layers. So that's 2 layers. Each of them is, of course, a phospholipid bilayer. So it's kind of interesting. I don't know really why it does that or why it's necessary, but I always thought that was kind of interesting how this membrane sort of folds in on itself. So it's keeping the DNA in. It's got an inner membrane, an outer membrane. Great names because it's easy to remember. And we have these nuclear pores. So let's talk a little bit about those nuclear pores, because of course, we have to let things in and we have to let things out of a nucleus. It's not just a closed structure, and that's what those nuclear pores are for. Here are some electron micrographs of those nuclear pores. They're called nuclear pores or nuclear pore complexes. You can use either term interchangeably, it doesn't really matter. And what you're looking at is, I can't remember how many proteins, but they're pretty large. I think it's 9 proteins, or maybe it's 10. I think it's like 1, 2, 3, 4, 5, 6, 7, 8, 9, something like that. And that's kind of what you're looking at. And there's a big hole in the middle. And these are not just open holes. These are very directed. And some things are allowed in and some things are allowed out. So it's kind of worth talking about this for a minute, right? Think about what is going into a cell and or into a nucleus and what is going out of the nucleus. Let's maybe start with what goes out of the nucleus, okay? So in the nucleus, everyone knows there's the DNA, okay? That's a given. Now the DNA is massive. It's not going to go out this little nuclear pore. But now what are you making in the nucleus that's going to go out? So in the nucleus, we are making messenger RNA. M is for messenger. Okay, so that messenger RNA is basically, there's a gene on it and it's going to go find a ribosome later. So that's important, right? We're making other types of RNA in the nucleus. tRNA is transfer RNA. We'll talk more about transfer RNA when we talk about translation, okay? So topic 15, we'll worry about that a little bit later. There's one other thing that we mentioned that we were making in the nucleus. We're making ribosomes, remember? So ribosomes actually come in subunits. There's a large subunit and a small subunit. And so they're not fully assembled until they're ready for translation. So the small subunits would exit independently and the large subunits would exit independently. So those are the things that are going out of the nucleus. So what is going into the nucleus? Well, we have to build all of these things. We have to build our DNA and our RNA. So what are the subunits of DNA and RNA? Of course, nucleotides. And we're also, in order to build DNA and RNA, we need enzymes. So we'll say enzymes involved in DNA and RNA. We'll just say processes because there's synthesis, but there's other processes that are involved in there. There are other things we could say. Well, we could write down those histone proteins, for example. And when you take more biology and you start to understand all of the functions that are going on in the nucleus, it starts to make sense of what has to go in and what has to go out. So those are the nuclear pore complexes. Like I said, they're very important for allowing things to go in, allowing things to go out, and they really are gated, meaning that things can't just go in and out. There is there are rules of engagement that allow the right things out and the correct things in. And without that kind of balance, you're looking at disease of the cell and sometimes disease of the body, of course, right? So we want to make sure we have the right things going in and the right things exiting the cells, of course. So one more thing about the nucleus. And this time we're zooming in and we're kind of looking at the inner surface of the nuclear membrane. And this is something that is basically holding the nucleus together, or at least the nuclear membrane. And this is something called nuclear lamina. So these are basically proteins, and they're fibrous proteins, and they're kind of like a scaffold. They're just supporting the whole thing. Then remember, the nucleus is a large organelle, very, very large. And without this, it would basically collapse. And so these are some semi-permanent structures just holding the whole thing together. So let's go and make some notes and about all these things here. Okay, so there's the nucleus, right? So the nucleus is a membrane-bound organelle. And we'll just say that it contains the cell's chromosomes. All right, so chromatin or chromosomes. So this is DNA plus associated protein. And we'll just say that the DNA contains the cell's genetic material or genes, whichever word you want to use. Okay, so hopefully those ones are no surprises to anybody. Then there's the nuclear pore complexes. So these are, don't want to use the pore in my definition, so we'll say these are holes in the nuclear membrane that allow materials in and out of the nucleus. Okay. What about the nucleolus? So remember, the nucleolus is a dark structure in the nucleus. This is the site of ribosome assembly. So basically, we were talking about a ribosome factory. All right, and the nuclear lamina. So this is a protein scaffold. And what does it do? It helps the nuclear membrane. Maybe I should say it supports the nuclear membrane. And that's the definition. Okay. Looking at this, I see the next one is ribosome, and we've already defined ribosome for bacteria, but I'm going to define it again, and I'll do that right now. So a ribosome is the site of protein synthesis. This has a name, it's called translation. Okay. All right, I'm just going to give you a moment to get all that down, and I'll say a couple of other things, and I'll switch back. And like I said, if you don't get all this, we will be coming back to it. And worst case scenario, you can always ask me to email you the Word document. Just send me an email, and I'll send it to you if you want the Word document. It's not really a big deal. What was the other thing I was going to say about this? Yeah, so all of these things here, right, are involved in the nucleus. The chromatin, the nuclear pore complexes, the nucleolus, and all that. A very important part of the whole cell. But eventually things start to move outwards, and we're going to start to have to make proteins and do all sorts of other things. And that's where the other parts of the endomembrane system are going to be important. So let's take a look here at some of those other parts of the endomembrane system. I am going to come back to this another day, the report tips. Like I said, I wanted to talk about the midterm today instead. So nucleus, nucleolus. Don't get them mixed up. One more time, ready? Nucleus, that's the big thing, contains the chromosomes. The nucleolus, that's inside the nucleus. So nucleus, nucleolus. Back to the endomembrane system. So you may notice that the nucleus is directly attached to the ER. The ER is the Endoplasmic Reticulum. So let's talk about that, shall we? So one thing on the endoplasmic reticulum are the ribosomes. And just showing you a picture here of the ribosomes. Kind of a cool picture. You can see they're made out of proteins and they're made out of RNA. So pretty cool. And I showed you this picture here as well to kind of highlight that eukaryotic and prokaryotic ribosomes are composed of different materials, or not different materials, sorry, they have different composition in terms of their RNA and their protein complements. And so the prokaryotic ones are 70S and the eukaryotic ones are 80S, so they're different sizes. And there is kind of a graphic showing the ribosome in action. So this here is the ribosome here, and much more on this later. What we're looking at is the messenger RNA feeding through it, and on the right we have the amino acids being attached together, and that is the process of translation. So like I said, stay tuned. I think that's topic 15, so much more on the functioning of these things later. So one thing that you may or may not know from high school is that there's really two kinds of ribosomes. Some of them are attached to the ER and some of them are not. And so sometimes we call these free ribosomes, the ones that are not attached, and the ones that are attached bound ribosomes. So I want to talk about the ER now and kind of get into what's going on there. So if ribosomes are making proteins, then clearly the ER is a big place of where proteins are being made. If you take a look, there's a transmission electron micrograph, and all of these spots, all those spots there, they're all ribosomes. And they're just packed in there. They're packed into the ER. They are loaded right there. So that's one thing to know about the ER, is it's a big part of protein synthesis. And in fact, it's a big part of the cell. In some cells, it's like half of the cell, quite a bit. Now, one other thing to know about the ER is that there's, it seems that there's two types of it. One that we call rough. So the rough ER is rough because it has all those ribosomes, and I can't spell here today. Those ribosomes, of course, make it look like it's got a bunch of spots on it. So that's why it's called the rough ER. The other part of the ribosome is the smooth ER, and it's kind of more tubular and has a bit of a funnier shape to it. So let's talk about those things there. Here's a kind of a cool cutout section. This is the, in the middle is an electron micrograph. And then we have kind of this artist cartoony drawing showing you what they think it looks like anyway. So I thought that was kind of a cool diagram. So what's going on with the rough ER? The rough ER is loaded with ribosomes. And so what does that mean? It means it's making proteins, right? So making proteins. In fact, we're talking about maybe, I don't know, maybe 80% of your cell's proteins. And most of these are membrane type proteins. And that's something we're going to talk about a little bit later on. It's going to distribute those proteins. Basically, it's going to send them to the Golgi. We'll talk about that in a moment. And it's also going to glycosylate these things. So this means adding carbohydrates. Adds carbohydrates. Hydrates to proteins. Okay, so I think I got a slide maybe for number 1, number 2, number 3. Maybe I don't. Maybe I'm going to come back to this in a later minute. So this is the big one, right? Making proteins. So remember that the ER makes proteins like crazy because it has all those ribosomes. So the smooth ER, there's a little cartoon about the smooth ER. The smooth ER is so smooth, you can see that he's being smooth, which sort of has that meaning in English to mean that, you know, he's able to talk to the ladies, I guess, is what they're implying. And whereas the rough ER is feeling disappointed that he doesn't get all the girls. Anyway, cute little cartoon there. I like my science cartoons. So what's going on in the smooth ER? So the rough ER is synthesis of proteins. The smooth ER, a big one, is synthesis of lipids. So you're probably thinking about, what lipids are we talking about? Well, in our bodies, we're talking about mostly phospholipids. Phospholipids. Because phospholipids are needed in every single cell. Now, there are other lipids out there in your body. Obviously, there's fat tissue and whatnot, and those kind of things are going to be made, and there's other lipids that can be made there, but kind of the big one is phospholipids. The other thing to say about the smooth ER is it has a lot of specialized functions in certain body systems. In your liver, it can detoxify drugs. in your muscles. It can store calcium. And those are very specialized. And I'm not going to get into those. Those are things you talk about in more advanced courses. Okay, so I'm hoping that most people have kind of seen a lot of this before in terms of the organelles, which is kind of why I'm going through this reasonably fast. If I'm going too fast, I'm sorry, just tell me to slow down sometimes or ask questions. That helps me to slow down. It's also very easy on Zoom to just, pardon my pun, zoom through the notes. Okay, moving on to the Golgi. So the Golgi body or the Golgi apparatus, or you can just call it the Golgi, is named after this guy here, Camillo Golgi. So he was staining cells and he saw something that looked unique, and eventually this organelle was named after him. So it looks like a bunch of flat pancakes or something like that. So let's talk about the Golgi. There's a micrograph of the Golgi there on the right, and the cartoon on the left. And you can think of the Golgi as, what do we do with those proteins we just made? Well, the Golgi receives them, it packages them, and ships them to the correct part of the cell. So let's take a look at what that means. So take a look at this, right? Here is our rough endoplasmic. And notice we have a little bit of RNA there. We're going to talk about these processes later on in the semester, but we've got that RNA, and we've got this little ribosome here, okay? And so the RNA, the sequence on the RNA is being used to make a protein. Now notice the protein here. This is the protein right there. Let's say that protein. So notice the protein is made on a ribosome that is attached to the rough ER. So the protein actually ends up inside the ER. We actually call that the lumen of the ER, if you're interested in that word. And eventually the protein is going to exit. So if you take a look, the protein exits in a little thing here. It's kind of like a tiny little membrane pouch, and that little membrane pouch is called a vesicle or a vesicle. You can use either vesicle or vesicle, it doesn't matter. Probably one is American, one is British. We're Canadian, so we'll just call it whatever we want. Yeah, so that little vesicle is going to move through the cell, and it's going to go to the Golgi. So notice the first part of the Golgi is the cis. It's going to travel through the Golgi, and it's going to exit in the trans part of the Golgi. So that's what it does. It goes to the Golgi processes a little bit, and then it's going to ship it somewhere. In the case of this one here, this particular protein is getting shipped to the plasma or cell membrane, and there it is. In fact, this protein here is getting secreted. So maybe I'll write that down. Secreted protein. There we go. And there it is. So that's kind of what it's doing. It's involved in protein secretion. It's also involved, and there's kind of from the textbook, you can see the same idea, starting with a rough ER. It's going this way through the cis Golgi, boom, 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 all the way to the trans Golgi, and then eventually out in the plasma membrane. So it doesn't have to go to the plasma membrane. It could go somewhere else. So you can think of it as a place where proteins, it's like the post office. Things get shipped out to other addresses. In this case, these addresses include other organelles, which is the back to the ER, the nuclear membrane. We could be ribosomes, vacuoles, all sorts of things. And of course, the one I just mentioned was the secretion. One more thing happens in the Golgi, and this is kind of a continuation of what was going on in the rough ER, is that the proteins get processed. And usually that means glycosylation. So remember, glycosylation means adding carbohydrates. And here's an example of a carbohydrate there on the right. You can see these blue things are N-acetylglucosamines. We have some mannoses and glucoses, and so on, right? And these carbohydrates are involved in cell recognition. I mentioned, I think before I mentioned blood groups are examples of carbohydrates attached to proteins, and the blood groups are helping your body recognize that those are blood cells. Okay, so maybe we should go and, before we get ahead of ourselves, go back to here and make some notes. So we talked about, we talked about all those things right there. Okay, so the rough endoplasmic reticulum, remember endoplasmic reticulum is ER, and so this is also rough ER, we can call it. And I'm not going to always spell out or say endoplasmic reticulum. That's a lot of syllables, isn't it? So what's going on in the rough endoplasmic reticulum? So this contains we'll say many ribosomes, and we'll say that this is involved in membrane, call it endomembrane protein synthesis. So meaning that it's making proteins that are ready and going to be targeted towards the endomembrane system or being secreted, which is in my mind all the same thing, because secreted is going to the plasma membrane, and the plasma membrane is part of the endomembrane system. The smooth ER is involved in lipid synthesis. And the example I gave was phospholipids. Okay, and there are other things that the smooth ER does, like I said, that are tissue specific, and I won't be asking you about, but things like storing calcium in your muscles or detoxifying drugs in your liver. In plants, they do lots of weird and wonderful things as well, but not going to get into that. That's kind of more advanced stuff. Vesicles, these are, well, what should we call these? Small membrane compartments, membranous compartments. And they transport proteins between organelles. There we go. And the last one there is Golgi. Or like I said, the Golgi is kind of like the post office of the cell. So this is, we'll call this shipping and packaging of endomembrane proteins. And there were kind of three things, right? We talked about secretion, shipping to other organelles, and protein glycosylation. By the way, I guess we forgot to add that for our rough ER, so maybe I will put that up there. Protein glycosylation. There we go. Okay, time check here. All right, there's a couple more organelles we'll talk about before cutting you loose for today. We want to talk about these last three: lysosomes, vacuoles, and peroxisomes. So that'll only take a couple of minutes here. So there's a little cartoon of the Golgi. Maybe we could take a look at this for a moment. So notice this is the Golgi here. So big kind of pancake-like thing, I guess. Maybe it doesn't look like pancakes on that one so much. Right in the middle, right there, we have big N, the nucleus. And notice those nuclear pores are probably a lot larger than they're supposed to be. All these little blobby things are vesicles. This here is supposed to be probably the rough ER. And in the background there, we have M for the mighty mitochondria. So more on mitochondria next day. So anyway, just a nice little picture there I found. Of course, there's all my writing. Okay, let's talk about the lysosomes, okay? So lysosomes are important as digestive compartments. You can think of them as cellular stomachs. They actually contain enzymes like your stomach, and they contain acid just like your stomach. So notice what is going on here. We talked about endocytosis. So there's some endocytosis right there. And you can see some food, and the food is going in. It's getting brought in by endocytosis. And then, so what happens next? Well, it turns out that now you have a food vesicle or a food vacuole, and it's going to get fused with a lysosome. So the lysosome contains those enzymes and acid, and now the food can get digested. So that's what a lysosome is. So lyse, by the way, means breaking, and zome means something like body or a portion or something like that. So it means a chunk of the cell that's involved in breaking things down, digestion. Cellular stomachs. Here's in the from the textbook, and you can see we have something being brought in by phagocytosis. So there's that word that we used in topic 4 and topic 3, topic 4. And if you take a look at the diagram, you can see the food vacuole being formed and the digestive compartments being made with an electron micrograph, the whole process at the top. So this is useful for eating food or destroying things if you're a white blood cell. Sometimes cells will use lysosomes for autophagy. So remember this word here, phage, phagocytosis, means eating. So this is self-eating. In this case, what they're doing is basically breaking down old and non-functional organelles. So you can see that very clearly in the micrograph at the top. You've got a peroxisome right here and a mitochondria right here getting digested in a lysosome. Okay, let's write that down. We'll just say a lysosome is a compartment for cellular digestion. Remember, it contains enzymes and acid. And actually, the acid is hydrochloric acid, so HCl. I'll put there. Okay, so what about vacuoles? Turns out we've actually talked about all of these different types of vacuoles already. Vacuoles are compartments and they hold stuff, and there's different types of vacuoles. So you may remember from lab 3, we talked about the central vacuole of the cell, and there it is right there. And this is basically in a plant cell, and it holds water and sap and things like that. There's an electron micrograph of one, and this can take up a pretty big chunk of actually a plant cell in many cases. We just talked about these food vacuoles, and so maybe we'll come back to that another time. And way back in topic 3, 4 again, we talked about things being in hypotonic solutions, and we talked a little bit about contractile vacuoles. So if you look where the arrow is, if you watch carefully, at some point you're going to see a circle, and the circle shrinks, and that's basically a contractile vacuole pumping out liquids. And just waiting for it to happen again. There, it's getting ready. Anytime now. There it goes. All right, so let's write a note for the vacuoles here. Vacuoles, we have 3 types. So food vacuole. Of course, I need to be able to spell. Let's try that again. There we go. Holds food from endocytosis or phagocytosis. We have central vacuole. Holds water and often sap and sometimes pigments in plant cells. We had contractile vacuoles. And what do? They pump out excess water. And if we're lucky, we might get a chance to see contractile vacuoles in the lab next week. We're going to be looking at this organism here is actually a paramecium, and we're going to get a chance to look at paramecium in the lab. I'm saying if we're lucky because sometimes when the weather's cold, we have issues with the shipping. I mean, they kind of, sometimes they get frozen in trucks and things like that. So I'm hoping we'll get a chance. Usually the paramecium are alive. Some of the other organisms I'm always concerned about getting frozen. Okay, let me just see where we are. I think actually what I'm going to do, just taking a look at the time, I'm going to actually stop there and let you go a whole minute early on Friday, so you can count yourself lucky. Anyway, I'm going to stop there. I hope you have a wonderful weekend. We'll see everybody on Monday.